This conference will now be recorded.
Good afternoon, everyone. I, Tabashri from the Science Book team, would like to welcome you all for today's session. Today's webinar on the era of stem cells and gene therapy using CRISPR-Cas9 technology is brought to you by the Team Science Web. Before starting the session, it's a humble request to all the participants, please keep your audio and video settings off in order to avoid disturbances during the session. Well, now I would introduce you all to our guest speaker. Ms. Mrunal Pande did her bachelor's degree in biotechnology from Fergusson College, Pune, India. Currently, she is a master's student studying molecular and cell biology at Ludwig's Maximilians University, Munich, Germany. Her research work is more concentrated on epigenetics, stem cell biology, and currently she is working on monoclonal antibody, DNA origami, and atomic force microscopy in a master's thesis. Furthermore, Ms. Mrunal Pande has done several internships in India from renowned universities like IISER Pune. I would like to thank our guest speaker for accepting my invitation and taking the time out of her busy schedule to join with us for this webinar. It's an honor and also a privilege for our team to host and participate in such a webinar session. Well, now I would request our guest speaker to take over. Over to you, Ms. Mrunal. Thank you.
Hi, can you hear me?
Yes, ma'am, you're audible.
Just okay, perfect. So I'll share my screen. Just let me know when my screen is visible.
Okay, so it's visible now. Your screen is visible, ma'am.
Okay. So first of all, welcome all of you to this amazing webinar on the era of stem cells and gene therapy using CRISPR-Cas9 technology. I'll just switch to the pointer mode. 1 second. Yeah. Okay, so in this webinar, I'm just going to like start from the basics of like what stem cells are, what is gene therapy, and what is obviously the CRISPR-Cas9 technology. And from there, I'm going to like dive into the recent research which like currently is going on. So before starting, so this is just the Instagram page. So if you want, you can just because I gave all of the updates of all the webinars and the recent research which is going on my Instagram page. So if you want, you can just go and follow. And also, I'm grateful for the Cyborg to give this opportunity to for me to give this webinar on this amazing platform. So without wasting any time, let's begin. So first thing is what makes us up, right? So if we take a human body, so we see like there are cluster of cells, right? So when we zoom in and we see the cells under the microscope, what we see is the nucleus containing the chromosomes. Now the chromosomes have genes, right? We like everyone knows this. As we are diploid, so we have two sets of genes. And when we zoom in the chromosome, we see the actual DNA. Now, what is the difference between a gene and a DNA? So, DNA is a genetic material, right? So, genes are basically the small segments in the DNA, and these segments they are responsible for the characteristics. For example, if a person has a brown eye color, or if a person has, for example, blue eye color. So, these are everything is governed with these genes. Now let's talk about the methylation. So methylation of DNA is basically an epigenetic modification. So if we see, like when we see like the DNA is in the unwrapped, it is unwrapped, right? So when the, for example, the DNA is methylated in the sense there is CH3 group which is attached to the cytosine, the fifth carbon of the cytosine. So we say that the DNA is methylated, and when this DNA is methylated, it tends to like fold and like kind of switch off the gene. So the methylation is responsible for the switching off of the genes. And there is one more epigenetic modification called as acetylation. So these acetylations basically involved in like switching on the genes or unwinding the DNA more. So let's talk about the methylation. So when we say that the DNA is methylated, it folds into a strand, okay, and then we have the nucleosome. So these are basically proteins which are involved in like winding the DNA around it. And when this nucleosome, it forms like a straight strand, and we call it a chromatin. And when this chromatin is more wrapped tightly around, then it forms a chromosome, right? So these are like the basic things which I need to clear out before starting. Now let's talk about the fertilization. So once the sperm
Ma'am,
and the egg meet after, yeah,
ma'am, good evening, ma'am.
yeah.
Ma'am, there is first slide is still shown only.
Ah, okay, okay. One second, one second. One second. I'll just reshare my screen. One second. Mm-mm-mm. Yeah, let me know. It's visible.
Yes, ma'am. Yeah,
Okay, I'll change the screen. Just let me know if it's changing. Yeah, I changed the screen now.
yeah. Well, it's functioning now.
Okay, perfect. So this was the second slide which I was saying, like we have the cluster of cells, and in that we have the nucleus, right? And when we zoom in more, we see the chromosomes, right? And then in chromosomes we have the genes, and genes, they're basically made up of like we have the DNA. And what are the differences between the gene and the DNA? So DNA is again, and the gene is basically the small segments in the DNA. And these segments of DNA are responsible for the characteristics like the blue eye color or the brown eye color, right? So these genes, they make up the DNA. Then the methylation of the DNA. So what I was saying is, like, if the DNA is methylated, what does methylation means? So CH3 is a methyl group. So when this CH3 is attached to the fifth carbon of the cytosine, we call it as a methylated cytosine. And when this cytosine is methylated, then this DNA can be folded more, and also the genes can be shut off. So if the strand is more tightly wrapped around, so this is done by the nucleosome. So these are basically the proteins. Okay, so these are like eight proteins, and they basically wrap the DNA around, and then we form a chromatin, which is like a strand of the nucleosome in which the DNA is tightly wrapped. And if we wrap this chromatin more tightly, we get the chromosome. Okay, so this was the basic concept which I wanted to clear. Now let's start with the next slide. So this is the fertilization. So when a sperm and the egg cell meet, we have the zygote, right? So 50% of from the father and 50% from the mother. So we have like two chromosomes. So it's a diploid. So the zygote, we say it's a totipotent cell. What does it mean? So the totipotent cell is a cell in which like the cell can directly form a whole another being. For example, if I say there are there are cells which are totipotent in plants. So it means that if I take one totipotent cell, I can produce a whole plant out of it. Okay, so that means it's a totipotent. Now, after the two cell stage, which means this one cell totipotent, when it divides, it forms two cells, and this is on the day one. We called it the two cell stage. Now, in the day two, the cells divide to form four cells. Okay, and after succeeding divisions, we get a morula, right? Where it's like the cells are still big, they haven't like changed the shape yet. And on the day 5 we get cells which are like dividing a lot, and also the size has become smaller. So this we call the stage as blastocyst. Okay, now if we take a cross section of the blastocyst, what we see is, so we have this outer layer, right? And inside we have this green color cells, not just green, I'm just, it's just one depiction. It's called as the inner cell mass. Now, this inner cell mass is where the pluripotent cells are, and this is what the research is going on. When we say, like, somebody is researching on embryonic stem cells, or somebody is researching on pluripotent cells, or they're studying some proteins. So these are the cells, like in which they're going to concentrate on. So what's happened is this outer layer is called as the trophoblast, okay, and this is the cavity. Okay, this is the inside cavity. It's called as the blastocoele. And then we have the inner cell mass, which is like joined at one end. And this inner cell mass, they are fully made up of the pluripotent cells. Now these are the cells, pluripotent cells, which makes up the germ layers, all the three germ layers. Okay, the endoderm, mesoderm, and the ectoderm. So pluripotent cells are responsible for making all the germ layers. And what does the germ layers make? They make the multipotent cells. What are the multipotent cells? So these are the cells which, when differentiated, give rise to like a specific cell types. For example, um, let's say like there is this hematopoietic stem cell. Okay, it is a multipotent cell. So these cell basically give rise to the RBCs. Okay, RBCs are the red blood cells. So these RBCs are total, like it's fully differentiated. So we call these the RBCs, or the for example, the T cells or the neurons, macrophages, everything. These are called as the unipotent cells because they cannot divide further, so they cannot differentiate more. This is like the end stage. So we call it unipotent. So these are the main terms. So we go from totipotent to pluripotent to multipotent to unipotent. So these are the four terms. Okay. Now let's see. So what happens to methylation of DNA and chromatin condensation during differentiation of stem cells? So like this is a question I wanted to ask you all. So what do you think? Like what happens? So when we say like when a DNA is getting methylated and if chromatin is going to condense, I mean it's condensating, then what do you think? Like when a cell is differentiating, does the methylation increase or decrease? So what do you think? Like the wrapping of the DNA. So if a cell is differentiating, like from stem cell, it is going to be an hematopoietic stem cell or RBCs. Then what do you think? Like is the methylation status going to increase or decrease? Anyone can answer. It's okay, I'll continue. So the thing is, it will increase. Why? Because when I say like a stem cell is differentiating, right? So differentiating means it's going to go under some changes where the stem cell is going to become like more what we say, it's into an RBC, for example. So RBC has a specific work, right? It has to carry oxygen and it has to like basically carry the hemoglobin. So transport oxygen, carry hemoglobin. So the stem cell is like, imagine it, like a clay. For example, if you take a white clay, okay, so this clay can be molded into different types. You can make a triangle out of it, you can make a rectangle out of it, a circle. Similarly, imagine stem cell as just a normal clay. It's not in random, it's like in random shape. And now you're going to take some clay and then you're going to like make a ball out of it or a triangle out of it. So we say that this clay, it's going to be like from like uh, so random shape is we are going to make a like proper triangle. So it means like in like a research, I mean more biological language, we say like the stem cells are getting differentiated into something very specific, because stem cells does everything. All the genes are activated in the cells, right? And in differentiated phase, so only some of the genes are activated and all the other genes, they are shut off. So we say that the when I say like the some of the genes are shut off, it means that the DNA need to be tightly rounded, and some of the genes need to be like like close off properly, so that it could be more, it could behave more like a red blood cell, for example. Because if all the genes, everything is on in the stem cells, then obviously it wouldn't function as a RBC, right? Some of the genes need to be shut down, and this shutting down is due to, I mean, it basically helps with the methylation. As I told, the methylation is basically what, when there is a methyl group added, then the gene basically shuts up and it basically starts winding together. So we have the DNA strand, okay, then we have the histone protein. So the DNA strand basically like encircles around it, it forms a chromatin fiber, right? So the all the histone proteins and basically the nucleosomes, they again assemble and they tightly round up each other and they form the chromatin. And from the chromatin we get when it again tightly wounds around each other, we get the metaphase chromosomes. So these are the chromosomes which is visualized during the metaphase. the methylation of the DNA and the chromatin condensation increases during differentiation of the stem cells. Okay, differentiation is basically specification. Now, what happens inside a differentiated cell? Okay, so these are some of the things. So the expression of the pluripotent genes. Now, when I say, like, the cell is going to be differentiated, it means that it's going to lose all of its pluripotency and it's going to become something very specific. For example, let's say neuron. So a stem cell is now going to become a neuron. So it means that the expression of the pluripotent genes, okay, the genes which are responsible for a cell so that it can remain in a pluripotent stage, it decreases, right? It makes sense, it has to decrease. Then second is the expression of the lineage specific genes. What is lineage specific? It means like now the cell knows, like, okay, now I have to become a neuron, or for example, now I have to become a red blood cell, so I have to now go from a hematopoietic stem cell path, right? So the expression of the lineage specific gene, it increases because now it knows, okay, now I have to be RBC. Then third is DNA methylation. As I told, like, the genes need to be shut off so that it could be more specific. It would work for something more specific. So the DNA methylation increases. And obviously the fourth, the chromatin condensation, it also increases, right? So these are some of the things which are going to happen inside a cell when a stem cell decides, okay, like, now I need to, divide and, be something more specific. This all is going to happen inside the cell once it has decided, okay, now I have to change. So what we saw was totipotent cell, which can form like a whole organism. Then we have the pluripotent cell, which will form all the germ layers. Then we have the multipotent cells, which will form all the like different types of, for example, the RBCs and everything, the differentiated cells. So these are some terms. Okay, so totipotent cells, so only zygote and the first cleavage of blastomeres, which means the two cell stage. Once one cell splits into two, we called it totipotent. So after that first cleavage is done, we cannot call it totipotent anymore. Then we move to the pluripotent, because once the cell splits, we cannot produce a whole organism. Then it becomes more specific. It will only go and form the germ layers. Okay, so totipotent, only zygote and the first cleavage of blastomeres forms all the lineages in mammals. Okay, then the second, now it divides more, and then it forms the pluripotent. Now what is it's the pluripotent cells are also called as the embryonic stem cells. So when you read some research papers, you have seen, they have mentioned ESCs or something. So ESC is nothing but embryonic stem cells. So they form most of the cell types, right? They form the germ layers. But one thing you need to remember is they form everything except the trophoectoderm. So what is the trophoectoderm that we saw? It's the outer covering, right? So we saw this stage when the cell was dividing, right? So after a certain time, we see like there is this inner cell mass and the outer layer. It's called as a trophoectoderm. So these cells, the inner cell mass, they are only called as the pluripotent cells and not the trophoectoderm. So this trophoectoderm is basically the outer layer of the blastocyst, right? We saw the blastocyst, which forms like during the day 8. So the outer cell layers of the blastocyst is important to form the extraembryonic tissue in mammal, right? And then we have the multipotent. Then it's the adult stem cells. It's also called as the adult stem cells, and it forms multiple cells of one lineage. For example, the hematopoietic stem cells. Okay, so multipotent stem cells example is the hematopoietic. Now this stem cell will make up everything, like the RBCs, the macrophages, the platelets, WBCs, the B cell, T cell. It will make up everything, but only of one lineage. Okay, so this is a
So,
multipotent. Then yeah. Okay. Then the differentiated cell, which is direct the last stage, right? Then we have the neuron forming, the epithelial cell forming, everything. So this is what a blastocyst looks under a transmission electron microscope, or the TEM we basically call it. So we have the blastocyst, right? You can literally see a cavity. And then we see a little blob here. It's called as the inner cell mass. Now these are basically the cells which are the pluripotent stem cells. Now these are the cells on which, like, when we take out the cells, we can do all types of research on them. For example, we can do epigenetical studies and everything. So these are the cells which where we are going to concentrate the inner cell mass. And then we have the outer covering, right? The trophoectoderm. So it's the outer layer. So this covering is the trophoectoderm. Okay. Yeah, and then this is again the extra. So this layer is the basically the zona pellucida, the ZP, which basically protects the inner cells. Okay. So what are the characteristics of stem cells? So these are like few main characteristics. They undergo organogenesis, which means they can form organs, which is true because like they are stem cells. Then they express telomerase. So telomerase is an enzyme. So when we see a chromosome, right? So chromosome is linear. We don't have a bacterial chromosome, right? If we see a bacterial, not chromosome, bacterial DNA, we see like it's a circular. But in humans, when we see our genetic material, it's linear. So the ends of the basically the DNA where it ends, we have telomerase enzymes which is acting at the end. So it what it does is it basically helps to protect the ends of the DNA. So as our like DNA is linear, so at the end it basically helps to kind of keep the ends. And this is where the aging comes in. So when we say like the cells are aging, it means that the telomere length is getting shorter and shorter, which means that the ends of the DNA are getting shorter and shorter. So once they get shorter, it means that a person will start to age. When we see, for example, if we compare like a newborn baby to a person who is like 50, 60 years old, we can really see, right? There are wrinkles, there is graying of hair. So this everything happens like due to the telomerase enzyme, right? Because it doesn't express that much in the adult cells. So when we talk about stem cells, we see that there is like increased expression of telomerase, which means that it will not go, I mean, the cells will not undergo aging. Then we have the extended telomerase repeats. So as I said, like it's a linear DNA. So at the end of the DNA, we have this continuous repeats. Why do we have this repeat? So that we can basically protect the genes which are important in our DNA. So these extended repeats, they are actually maintained and added up with the help of the telomerase, right? So they have extended telomerase repeats. Then obviously stem cells can differentiate. They can form so many cell types. So they have the ability to differentiate. They can be tissue specific. Then they undergo apoptosis. Apoptosis is basically spontaneous cell death. So if the stem cells think, okay, it's not behaving properly, or if it thinks like it's going to become more cancerous or something, then that's the time when it decides that, okay, now I have to like shut down everything. All of my machinery should be just shut down, and I'll just go and just break off. So this is when the apoptosis comes in. So the stem cells can undergo apoptosis. That is, it can literally induce like cell death. Then it can migrate. Obviously stem cells can migrate from one place to another. Then they are heterogeneous with different phenotypes, right? Then they have this self renewal capability. So when I say like stem cells are differentiating, it doesn't mean like the cell will directly differentiate. No, first thing is it will do a self renewal, so that a certain threshold is never it's not lessened. For example, if there is one stem cell, it will divide into two stem cells, and the first stem cell will undergo the differentiation. So the one stem cell is always maintained at the top. Then again it will divide into two, and the another will undergo differentiation. So always a threshold is maintained, and then it will proliferate indefinitely, of course. So it has like a higher proliferation capacity. Then if we look at this, so what we saw is the sperm and the egg meet, then it forms a zygote up to like first cleavage. It is called as a totipotent. Okay. Then after that, when it becomes a blastocyst, right? We saw the blastocyst stage. It's a fifth day stage. When we see the blastocyst, it becomes pluripotent. Now what we do is like for example, if someone is working on the embryonic stem cells, what they do is they'll take this blastocyst. For example, if we want to work on human stem cells, right? So what we do is we take a blastocyst from human. Okay. We take this inner cell mass, right? So these are the inner cell. This is basically all the pluripotent cells which we saw, and we will take it and then we will plate it in in basically a embryonic stem cells plate. So I'll kind of discuss it in further slides. And then this is the pluripotent cells, and then we can do whatever research or what we want to work on. But now if we don't do this, if we let the blastocyst to differentiate more and then we extract the cells, we will get all the multipotent cells which are like very specific, right? You'll get the neural stem cells which will only form the neurons. You'll get the hematopoietic stem cells which will only form the blood related cells. You have the cryptic stem cells, which is of the gut or the intestinal lining. Then the bulge stem cells, which will form the hair skin cells. So it will be very specific. So what you need to like pay attention to here is like the when we go from totipotent to multipotent, like from up to down, we see that the expression of the pluripotent gene is going to reduce, and which makes sense, right? Because now we are not going to be pluripotent, we're going to be multipotent. So the expression of the pluripotency genes decrease. Then the expression of the lineage specific genes. Lineage is like the neuronal stem cells, they have a fixed lineage that it can only form the neurons. So this expression will increase. Then there is the open chromatin. Okay, so the open chromatin will now be packed up tightly. So it will not open anymore. So it will decrease. And the DNA methylation. So the DNA methylation will increase as the stem cell starts to differentiate. The methylation also will increase. Then, what is the difference between the adult stem cell and the embryonic stem cells? Okay, it doesn't mean like this is okay. Anyways, so what is the difference between adult stem cells and embryonic stem cells? So these are the cells which are undifferentiated cells, and it is found throughout the body after the development. And these cells are essential in the daily healing, growth, and replacement of the cells which are lost. And the embryonic stem cells, so these are the cells which are derived from the early stage of the human development, which we saw was the blastocyst. And they are pluripotent. I'm sorry. So they are basically pluripotent, meaning like they have the potential to differentiate into any cell type of the body. Then characteristics, so they basically enable the adult stem cell, they basically enable healing, growth, and replacement of the lost cells. Then they're isolated from the adult tissue, such as bone marrow and fat. Okay, so basically these cells, they are not more plastic. Okay, plastic in the sense it's like they are not very flexible, they are very lineage oriented, like I know that I have to become a RBC, so I'll become that. But embryonic stem cells are not like that. They can become RBC, they can become neurons, I mean they can become whatever they want. So this is the basic difference. Then they are used for regenerative medicine and tissue repair. Then there are fewer risks of tumor formation compared to the embryonic stem cells. And for the embryonic stem cells, the use of the embryonic stem cells is controversial and raises ethical concerns, because obviously we are going to take embryos from humans, right? So it's a little obviously ethical questions arise. Then embryonic stem cells have the ability to differentiate into any cell type, right? And it has a high rate of cell division. Then the thing is like the ESCs, or the basically the pluripotent cells, they undergo many cell divisions. Okay, it is easy to obtain, but the blastocyst is destroyed in the process. So when we need to get out the inner cell mass cells, but the blastocyst gets destroyed. And the adult stem cells, it is less stable, so the capacity for self renewal is limited. Okay, but it is difficult to isolate from the adult tissue, because we are going to work on bone marrow, so it's really difficult to get the stem cells out. Then how are the stem cells maintained in the body, right? So the embryonic stem cells, or the pluripotent cells, they have these special transcription factors and co-regulators, which maintain the pluripotency stage. So oh god, okay, just I'm really sorry, just give me 1 second, I'll just go and check. Hi, okay, sorry for that. So when we say like a stem cell, so basically what are the basic inner mechanisms which makes a cell behave like a stem cell, right? So these are like the three main genes which are responsible for the cell. So once a cell express these genes, it means that the cell is pluripotent. Okay, so we say like, okay, so through these interactions, okay, their interactions with various partners, so obviously the genes, they form proteins, right? So we have the DNA forms the mRNA, which is called as a transcription. Then from the mRNA it forms the basically the proteins. So we have the translation process. So these all genes, they basically make up the proteins. So these are the nanog, oct4 and sox2. So these are the three genes which are responsible for the cell to behave like a stem cell. So these basically the proteins, they interact with various partners and they activate the transcription of the pluripotency elements and repress the transcription of the lineage specific elements, right? So we need, when we want like a cell to behave like a pluripotent cells, we want the transcription of the pluripotency elements to increase, and the lineage specific elements like the cell should undergo differentiation, so that part should be repressed, right? Because we want a cell to behave like a stem cell. Now, the what is nanog? Nanog is also called as the homeobox or transcription factor, and SOX2, it's also called as the SRY box trans factor 2, and then we have the OCT4, which is octamer binding trans factor 4. So these are like the three main proteins. So what they form is they basically form autoregulatory loop to control the expression of each other. Okay, the responsible for inactivation or repression of the lineage specific genes or elements, and then they form the cross regulatory loops as well to activate the expression of the pluripotency associated elements. Now what does this mean? So basically, so if I don't know if you have like read about this somewhere, but we have integrins, WNT signaling pathway, and then we have the BMP4. Okay, so these are basically the signaling pathways which when activated, okay, which when activated, they basically like open up a whole cascade under the cell. For these are basically like they're present on the surface of the cells. So once this, what we say, this integrins or WNT, they are basically activated by molecules binding on it, then it will basically communicate inside the cell to open up a whole signaling cascade. So these will activate the beta catenin, the SMADs, the IDS. Okay, and after activating this, these all will go and activate the OCT4 and SOX2. Okay, so one thing you need to remember is that so OCT4 and SOX2, they dimerize. What does dimerize? So it basically they interact with each other, and this interaction will lead for nano to forms. I'll basically explain for the slides. Yeah. So the interaction of different proteins with OCT4. So the OCT4 is not only going to interact with the SOX2 and NANOG, but there are like whole lot of proteins you can see. So the OCT4 participates in these first cell fate deciding events. What does it mean? So if the OCT4 is less, okay, the amount of the OCT4 protein is less in the cell, it leads to different or inappropriate differentiated process in the embryonic stem cells. And if the OCT4 protein is more in the cell, it means that it leads to the differentiation into primitive endoderm and mesoendoderm. Now it means like if there is a lot of OCT4, then the stem cell is going to differentiate. If it's less, then there are not going to be a lot of differentiation process. Here we see the obviously the blastocyst. So we have the inner cell mass, and then there will be an inappropriate differentiation, which means that it wouldn't know, like, what I have to be. So this is basically when there is loss of OCT4, like there is very little concentration in the cell. And now if the OCT4 increases, okay, then this inner cell mass, it will start differentiating into primitive endoderm and mesoderm. So these are basically the germ layers, right? It will start differentiation once the concentration of the OCT4 increases. Now, the nanog expression. Now, increases in the cells in the inner cell mass. This gives rise to the pluripotent epiblast-like cell. Now, what is this? So, we first have the morula thing. Then we have the inner cell mass. You can see the purple color is the inner cell mass. The green is the outer layer, the trophoectoderm. So once it reaches certain stage, some of the inner cell mass, you can see it will start to differentiate into epiblast. It's also called as epi, so red color ones. So some of the cells have started differentiating into epiblast like cells. So these are the cells which will start to form the germ layers. So now this after this cell mass, it will go and attach to like one corner, and then after that the cell's shape will change, everything. So the inner cell mass will differentiate into two types. Some of the cells will go and become epiblast, and some of the cells will go and become the primitive endoderm. So you can see here, so this is the primitive endoderm, and now all of the trophoectoderm, it will start to like go inside, right? So this is the movement, so this is how it will start to go inside. And the primitive endoderm, so the purple thing, okay, the primitive endoderm will start to form the visceral and the parietal endoderm. So these are the two types of basically the endoderm. You don't need to go in detail. So what we see is, so this types of cells, when they start to differentiate, they form the epiblast. Okay, so epiblast is this inner cells, then the hypoblast, which forms the line, and then we have the trophoblast, the outer thing. And this happens on the day 9. And the what we saw, the blastocyst, it was the day 5, and this happens in day 9. So what is the epiblast? So epiblast, also known as the primitive endoderm, because it forms the primitive endoderm, it is one of the two distinct layers arising from the inner cell mass. And specification of the epiblast as a cell lineage in early development, so it is basically a specification that, okay, if the epiblast cells is present in the basically the blastocyst, then the early development is coupled with that of trophoblast and hypoblast, and two lineage dedicated to form extraembryonic tissues. So when we once we see like, okay, this is day 5 and this is day 9, so you can see that the inner cell mass, it has started differentiating into epiblast cells and primitive endoderm. So two types of different cells are formed. Now the cells have understood, okay, now what we have to make, right? So the epiblast cells and the primitive endoderm. And now the primitive endoderm elements, they are basically repressed, and the lymph or the stat, so lymph, I'll discuss this in the further slides, that independent of self renewal of pluripotent ESL, I discuss this part in the further slides, but this is what happens. So in the day 5 and day 9, then what is the primitive endoderm? So it's basically, it's a cell lineage segregated from the epiblast in the blastocysts, and it gives rise to the parietal and the visceral, this one. So parietal endoderm, and the primitive endoderm, it splits into parietal and visceral. That's what is written. So what happens is, when we say like it splits into this, finally expressed into primitive endoderm, the SOX7, is basically the expression of the SOX7 increases. So this is like amazing picture I got on the net. So we can see, like, in the inner cell mass, you can see the OCT4 protein, GATA6 protein, and nanoc, they're expressed at high levels. And in the trophoectoderm, the CDX2 protein is expressed. But if the OCT4 expression goes down, now the cells know, okay, now I have to differentiate, because the OCT4 concentration has gone down, and only the nanoc concentration will be there. Nanoc is a protein, right? So it only the nanoc protein will be there. Now they will form the epiblast. If GATA4, SOX2, and DAB, all of these proteins are expressing, then the cells know, okay, now I have to become a primitive endoderm, right? And then similarly for the other. So what happens is nanoc. So nanoc expression, it's basically, if it's expressed, what it does, it is inhibits the primitive endoderm element. So what if the nanoc is present, then it will not form the primitive endoderm, right? It's going to form the epiblast, and it activates the live start pathway. I'll explain the further slides. Then if the nanoc is not there, for example, it's very low concentration of nanoc, then spontaneous differentiation will take place. If a little of the concentration of the nano goes down, it will start to differentiate into primitive endoderm. And if there is overexpression of nanoc, then it will promote self-renewal independent of the LIF, which is the leukemia inhibitory factors, which are basically secreted by the epithelial cells. Now here you can see, like, it's a, it's image under the transmission electron microscope. So you can see, like, trophoectoderm. So this outer layer basically forms the placenta, okay, and this is the inner cell mass which forms the fetus. Then now you had like glimpse of how these, so we know, like, proteins are interacting with each other and everything is happening. So so this is like how a unified network looks like. So these are the green ones, these are basically the genes associated with self renewal and pluripotency. What does it mean? So it means, like, if these genes are activated, then the stem cells will start replicating, that basically they'll have a self renewal capacity, and also they'll become like basically pluripotent. Then if and this is depending on the regulation of this. So this is the main thing. So their own and each other regulation and expression is highly coordinated in a highly coordinated manner. So SOX2, OCT4, and NANOG. So these are the proteins which, depending on their concentration, the cell is now going to be either stem cell, or either now it's going to go to the lineage commitment, or basically it's not going to get differentiated. So everything depends upon these three proteins. And then these are the repression. So if these genes are repressed, then the expression of genes in they are involved in the lineage commitment. Okay, then let's move. So cells. So this is basically the Waddington landscape model. So this I found during one of my lectures. So this is basic. Imagine like there is a ball. Okay, so like if you keep a ball here, okay, and if you can kind of leave the ball will like come down. So there are two possibilities. So it can either go to the left side, or it can either go to the right side. Now it depends, like depending upon the the push it gets, it depending upon, and the turn, it depends upon like where it's not going to go, right? So it's similarly with the stem cells. Now if we leave the stem cell, depending upon the protein concentration, how much is it, like what like how the regulation is there, depending on that it's now going to be like differentiated or not differentiated, like that. Similarly, in the right hand side, you can see there is an embryonic stem cell, or the inner cell mass cell, basically. So they have everything. They have the SOX2 protein functioning, they have OCT4 protein functioning, they have the NANOG functioning. And when you leave it, what you see is like now they have more of the SOX2 protein forming. And if the cell has more of SOX2 like protein, then it means that it's not going to get differentiated into neural ectoderm. And if has OCT4 protein functioning, then it's going to form the mesoderm, like that. Then the cells, they have also have the ability to trans differentiate. What does it mean? So one cell can turn into another. So it is also called as a direct reprogramming. So like the cells can like kind of change from like one form to another. So that is also possible, trans differentiation, because they still are, that they have a little plasticity in them. And when I see, like a stem cell is getting differentiated, then epigenetical changes play a huge role. Epigenetical changes means the methylation of the acetylation of the DNA, there is sumolation of DNA, there's so many things. So all of these epigenetical changes will take place. There are histone modifications. Then all of these changes will play a huge part when I say like a stem cell is now going to differentiate into for example, any cell. Then just a recap. So homeodomain transcription factors. It's also OCT4 is also called as the POU5F1. So it maybe in some papers they mention OCT4 as POU5F1 and NANOG. And then we have obviously the SOX2 gene, and they basically form the transcriptional module that has a central role in maintaining the embryonic stem cells. So if these are properly functioning, then a cell can be in a stem cell state, or it can start differentiating depending upon the concentration. Then they have reach positive feedback loop, which means that the product, whatever the product is going to form, right, or the proteins are going to form, it can lead to the increase in the reaction. For example, like if I say like the gene is activated and now it's forming a lot of nanog. So like more nano concentration in the cell will activate the gene to produce more nanog. So it's like a positive feedback. Then the feed forward loop. So it consists of three genes, that as I told you, these three genes which code for three different transcription factors like A, B, C, and B regulates C and A regulates both B and C. So like you can kind of imagine it, when you like draw it, it would be like a little easier rather than this confusing it. And then we have the OCT4 and the SOX2 form heterodimer that positively regulates the expression of the POU5F1. Oh my god. Yeah, and then we have the SOX2 and the NANOG directly interacts with the OCT4 and positively regulates the expression of all the three genes via positive protein-protein and protein DNA feedback loop interactions. So this is a little complicated. It would be easier if I have like included diagrams, but okay. Yeah, all three genes activate numerous developmentally important genes and repress the expression of the genes involved in the lineage commitments, which we saw basically. The loss of expression of these genes leads to loss of self-renewing embryonic stem cells phenotype, and it leads to commitment to differentiation. Then, so how to isolate these stem cells? So we know that, okay, fine. Now this is the late blastocyst, where the inner cell mass, it has differentiated into epiblast and primitive endoderm, right? So primitive endoderm is from where basically you get the extraembryonic endoderm stem cells. So these are, if you basically extract this layer, okay, the primitive endoderm, you will get the extraembryonic endoderm stem cells, or the XEN. So if you have read some research paper, you might have come across this term. Then if you extract the trophoectoderm, the outer layer, you will get the trophoblast stem cells, okay, the outer layer. And now if you extract the epiblast cells, then you will get the embryonic stem cells, ES, or the epiblast stem cells. It depends upon on which day you extract, right? So here you can see exactly. So what we do is we remove the inner cell mass, then we grow it in a dish, and now this can differentiate into epithelial or neural cells. So this is what the it's very basic process which I'm showing here. So so which conditions exactly do we need to drive differentiation of these cells, right? So if we take inner cell mass into a plate, now what can we change in the plate? Like what type of medium can we use so that the cells can now start differentiating? Okay, because you need a certain environment, right? The cells need to be happy in the plate, right? So when we say like a cell, now we want the cell to differentiate, so we need to change something in the environment of the plate so that the cell now will start differentiating into epithelial and neural cell. So the culturing of the stem cells in the lab. One second, I just drink water. Yeah, so normally what happens is, like, when we talk about the stem cell media, we have the DMEM, okay, then we have 16% FCS, which is it's not basically always mandatory, you can add it or not. Then we have the LIF, which is the leukemia inhibitory factor. I'll talk it in the further slide. And also the 2I. So LIF and 2I are the main thing, right? So you can see here, these are like two different like conditions in a plate. So what we do is, these cells, you can see these are different type of cells, okay? These are called as the feeder cells. So we take a plate, we put like a cells on the surface. So if, for example, here, so we take a plate, we put cells. So on this, like the bottom layer, we have feeder cells. So you can see this lines, right? So these are the feeder cells. Now these cells are basically going to secrete LIF. What is LIF? It's leukemia inhibitory factor. Now if this LIF is secreted, it means that the stem cell will remain a nice good stem cell, okay? It will not differentiate. Now what is in the second scenario, what we see is, this is also embryonic stem cell colony, but we are directly adding LIF, okay? There is no feeder here, we're directly adding LIF in it. And you can see the difference, like the colony has a sharp borders, right? You can see, like, okay, it has a certain shape, but here you can see it's a diffuse border. It looks like an amoeba basically. So here you have a defined borders, and here you have undefined borders. So what should be like a good colony? When we say, we are looking at the colony, what, like, how should it look like? So this is what the colony is. With what we need is, we need sharp borders but with no feeder layer. So this is what we want. So the thing is, like, if we add LIF, okay, disadvantages of LIF over feeder cell replacement is that it will diffuse the border of colonies. Then the embryonic stem cells turn out to be more primed. So when I say, like, when I take the inner cell mass into the plate, okay, so these cells will be very plastic, okay? They'll form whatever you want them, they will form. But if they are not maintained properly, if the stem cells are not maintained properly in the plate in the lab, then the cells will turn like into like very rigid structures. For example, it's not naive anymore. Naive, it's like you can turn the cell into any cell type, but now it is not naive, because you are like, it's not maintained properly. Now it will differentiate. It's like, it's going to be little differentiating into more primed stage. We say it's like more primed. It's less flexible, it's less plastic, so it's more primed part of the cell. So the primed cells differentiate much quicker, okay, in culture than naive cells. So once the naive cell changes into primed cells, it will start differentiating, okay? So this thing, so that's why we need the cells to be in the naive stage. And One second. Yeah. So the naive or the prime cells. So you can see this is the human pluripotent stem cells. So these are the primed one. So what happens is the primed one, you can see these are markers. Okay. So you can see this cell, it's glowing green. It basically shows like the X chromosome reactivation has started. So we know that, for example, a boy, he has basically like X and Y chromosome, and a girl has obviously two X chromosomes, right? But does if like how can we say like, a guy is like performing well with just one chromosome, one X chromosome, and girls they have two X chromosomes. So the thing is like we need to compensate somewhere, right? So either one X chromosome of the females should be like shut off, right? So that it could be equal to the guy's X chromosome. Okay. So what happens is there is a doses compensation in females, where what happens is, so we have two X chromosomes, so one X chromosome is always shut off in females, so only one X chromosome remains functional. Okay. So this is important for so that we don't get duplexes of protein. For example, if my body need like some, for example, random, let's say protein A, so my body need like only 10 protein A's. If I have two X chromosome which are activated, then it will have like 20 protein A functioning, right? So it will be double. So we need to have a doses compensation. So what does our body do? The human females, they should always have one chromosome which is shut off. So what happens is when we say like a prime human stem cells, if it's getting back to the naive stage, then what happens is the X chromosome reactivates. So one X chromosome is shut off, so it starts to reactivate now, because naive is like the early stage. So all the markers are going to be basically activated, and the primed marks, so primed is basically as I told you, it's naive is basically less, I mean it's more plasticity, and the primed is basically very it's not plastic anymore. So the prime markers are going to go down and the naive markers are going to increase. So this is what's happening, and the DNA methylation level will also go down because the cells are becoming naive. And if you want to work on naive, then like this is kind of nice. Then this is I took this from a paper. So this is what primed cells look under a phase microscope, and this is what looks at in the GFP channel of the microscope. So this is a phase channel and the GFP channel. So they have basically, what they have done is they have marked the GFP to the OCT4 protein. So in naive mouse ESCs, a particular enhancer of the gene OCT4 is active. So we can see that in naive cells, this OCT4 gene is activated. So you can see here, right? You can see the cells. So you can see the OCT4 gene is activated. So the prompting the researchers to look for the presence of this markers. So it means like if the OCT4 gene is present, then the cells are naive cells and not primed cells. We can differentiate between these. So if you have cultured your embryonic stem cells for quite a while already, and now you want to know whether your cells are primed, then why would you want? So first question is, how could you check whether your cells are naive or primed? And second, why would you want to know? Does it matter if it's naive or primed? And why is it important to know whether your cells are naive or primed? So the thing is, first we can check the cells, they are naive or primed by, obviously, first thing is that we can observe under the microscope. So the morphology of the naive stem cells, it's much smaller than the primed cells, right? Then also the naive cells have diffused boundaries and primed cells have outer sharp edges. Second thing, naive stem cells are derived from the inner cell mass of the pre-implantation blastocysts. So this is like a major difference. So the thing is, when we see a blastocyst is forming, after some days, the blastocyst will go and attach to the uterine wall, right? So once it goes and attach to the uterine wall, we say it is either it's pre-implantation or post-implantation, right? So these are the two stages. So when and it has not yet attached to the uterine wall of the female, then this pre-implantation blastocyst, it will have all the naive cells, and once it gets attached to the uterine wall, it will have all the primed cells. Primed is basically, now it knows that it has attached, it will start differentiating now, like that. So these are the difference between the main difference. So it is important to know if our cells are naive or primed, because primed cells differentiate much quicker in the culture rather than the naive cells. So if you want to do some research in the stem cells, and suddenly next day you come and see your plate, like your plate has already changed, the cells are already changed into something else, then you should understand, okay, the stem cells have already differentiated and they are not stem cells anymore. Yeah, so we have this leaf which I said. So this is like a normal cell culture media. What we need is there is attachment factors. Now what are these? So when we put cells into the plate, we need the cells to attach to the bottom, right? So these are the attachment factors. So cells have the tendency to go down to the plate and attach. When we see a mammalian cell culture plate, you can literally see in the light, like you can literally see a layer of cell at the bottom. Then we have some vitamins, we have glucose, inorganic salt, there is pen strip is penicillin and amino acids. Then we have the buffer and we have the serum, of course, for the growth factors and hormones. So you can see, so we have the knife cells. Knife cells change into something in between, which is like the formative, and then we change into prime. So these are the proper embryonic stem cells, and then they turn into something primed. Primed are also stem cells, but now they know they are going to differentiate. And now what? Now we are going to add LIF in the media. What is LIF? LIF is basically it's normally expressed by the tophoectoderm, which is the outer layer of the blastocyst, right? Of the developing embryo. So they have the receptors called as the LIF-R, which is expressed throughout the inner cell mass, so the inner cells. And as embryonic stem cells are derived from the inner cell mass at the blastocyst stage, removing them from the inner cell mass also removes the source of the LIF, right? Obviously, it makes sense. Then the leaf is often added to the stem cell culture as an alternative to the feeder cell culture, because when you look at the microscope, it's really it's hard to see, it's whether, for example, in this picture which we saw before, it's really difficult to see which I mean this colony is nice, so we can see, but if the colony is not well formed, then we cannot say if it's a feeder layer or if it's a feeder colony forming or if it's our colony. So it's better, like if we add LIF, then there is no need for a feeder layer, we can just add LIF in the medium. And then it regulates the embryonic stem cell self renewal, which is good. Then it is involved in the JAK signaling pathway. It activates the make signaling. These are basically a different signaling pathway, which is important for a stem cell to be a stem cell in the plate, right? So, and yeah, then it activates the STAT3, and then it activates, no, STAT3 indirectly activates the pluripotency associated elements. Okay, then what we add, so with this leaf, we're also are going to add the 2I. What is 2I? 2I is basically called as 2 inhibitors. So this 2I media, they're going to have 2 inhibitors, which is GSK3 beta and MEK 1. I mean, this like 1, 2. So these 2I basically helps the cell to, so so wait 1 second. So here we saw we need a colonies with sharp borders but no feeder layer, right? So something nice, this, but no feeder layer. So this is what basically 2I does. So it basically gives a nice homogeneous naive cells. But if you don't add 2I, or if we only add LIF or serum, then the naive cell will turn into primed, and once it become primed, it will start differentiating. Okay, and here you can check the epigenetic status. So as the methylation level increases, then it will start differentiated. If it's low, then it will be a stem cell. Okay, and obviously you can check the expression distribution. So here these are like SNAI ones. These are basically different markers of the gene expression. So we see like if 2I is added, is required. So this is like one of the things which we put in the media, which is really important if you want to culture a stem cell. So now, how to induce differentiation in culture cell models? So we have pluripotent cells. If we remove LIF or 2I from the medium, the pluripotent cells will start differentiating and it will form a embryoid body. It's also called as the EB. Okay, it will form something like a structure like this. So there is this method called as the hanging drop method. So these are basically used when you want to do research on some embryoid bodies. So it's like this is day 0. So you put embryonic stem cells, you take the embryonic stem cells, you prepare hanging drop, and you like attach like I mean at the end of the drop you have this embryonic stem cells, embryonic bodies, and then you can directly cultivate it in the suspension, and this is basically how it looks like. And yeah, so undirected differentiation, potentially it forms all the three germ layers, so whatever you want. So endoderm layer, ectoderm layer, mesoderm layer, it will form everything. Okay, so if you want to make some neuronal cell, or some thyroid cell, then you can make with help of this, only changing some protein, I mean the contents in the media, you can differentiate it. Then so this is basically how it looks under microscope. It's really beautiful because we are going I mean we tag like a lot of proteins here, so we use GFP, DAP, there is I guess RFP also being used. So yeah, we are going to tag it with different markers. So we have embryonic stem cells, which turns into embryonic body after differentiating, and then from this embryonic body we can isolate everything. We can get cardiomyocytes, neurons, hepatocytes, these are the liver cells, pancreatic cells, this is heart cells, skeletal, we can everything. Then the epiblast. So basically, when the inner cell mass, when there is methylation modification, then it forms the epiblast cells, which we saw. So it's just like a recap thing. So this is one of the in the research paper I saw that. So there is this DNMT3B. So it is basically what do you say? It's like a methylation transfer enzyme. Okay, and it's basically responsible to load the methyl onto the DNA. And when the methyl is loaded on the DNA, the gene will shut off. Okay, so this DNMT3, so they saw the localization through the differentiation. So what they did was they tagged this DNMT3 protein with some, let's say for example GFP. Okay, and now what they saw was like when the cell is not differentiated, when it's in the stem cell stage, there is no expression of the DNMT. As soon as the cell has decided, okay, now I will start differentiating into something very specific, the DNMT3 level will go up, right? And once it go up, you can see in the photos like there will be like a certain spots. So this DNMT3 will be only working on the DNA, right? So you can see the like spots in the cell, so it's working in the on the DNA basically. So and at the end you can see the differentiation, you can see a lot of like all the cells are basically bright, and these are the cells under microscope. So you have the totipotent blastoma, epiblast, and then yes,
हट
we
जाओ
have yes, medium, which consists of the DMEM, LIF or serum or 2I basically. Then we need anchorage. For example, as I said, the cells need to attach, right? So what we do is when we take a new plate out of a bag, we put gelatin, we pour some gelatin over it, and we keep the gelatin for 20 minutes. And after it's done, we take out the gelatin. Now the cells can, when we put the cells on the cells can go and attach to the gelatin. So the gelatin is important. We basically use it in the lab. Yeah, yeah, so these are like one of the most important point. It's like there are niches in of stem cells. For example, there is only a certain place where there will be I mean the stem cells will be present. For example, if we talk about bone marrow, there will be a niche in the bone marrow where the stem cells will be present. And once I mean the niche is basically environment, right? So for example, if I feel comfortable in this environment, then I mean obviously I live here. But if I think like there is a lot of stress and there is there is sound and everything, so I leave this house and go live somewhere else. So this is basically a niche. So niche is where the stem cell is really happy, it's getting everything, it's getting all the nutrients. And now like there is a lot of stress signals, or or the stem cells have decided itself like, okay, now I need to differentiate. Then it will leave the niche and then it will start differentiating. So we have these different niches in the bone marrow as well. So we have two types of niches. We have the quiescent storage niche and we have this self-renewing niche. What is quiescent storage niche? So when we have stress, when we have like work stress or something in our life which is like stressing us a lot, then our stem cells will undergo this stress response and it will start to differentiating. So like if you have seen like the people who have like a lot of stress, they start aging faster. If I don't know if I've observed that, like they'll have more wrinkles and everything. Like the aging will be much more faster than the people who don't have this stress thing. So this is what happens. Then we have the second, it is self-renewing niche. So what, as I told you, right? So the stem cell will split, then only one of the stem cell will go and differentiate, the other stem cell will be there. So it will basically like it will always maintain a threshold in the niche environment. So there will always be stem cells in the niche. So this is the self-renewing niche. Then which adult stem cell to be, this is just an example thing. So yeah, so mesenchymal stem cells, it forms the bone, cartilage, tendon. We have the oval cell which forms the hepatocytes, these are the liver cells basically. Then we have the cardiac progenitor which forms the cardiac myocytes, which is the heart cells. Then we have the hematopoietic stem cell which forms all the blood cell types, like macrophages, then platelets, RBCs, lymphoid, everything. Then this is some recent research which I found. So they recently, so it's just August 30, right? I guess it's 2nd September today. So on just August 30, they found there are stem cells in our thymus gland. So here, near our throat. So yeah, so they found that there are stem cells in the thymus as well, in humans. Then second is like they form that cells have been artificially reprogrammed into states which are similar to the embryonic stem cells, and these are known as the induced pluripotent stem cell, and they can bear a memory of their previous history. For example, if the cell, if there is some random, let's say if there is a skin cell, okay, the skin cell is already differentiated. Now we want our skin cell to go back being a stem cell. So what we are going to do, we are going to provide some factors and it will now change into a stem cell. Now this stem cell will have a memory of its previous history. Like when I was differentiated into a skin cell, it will always remember that. So they found something like, it's really recent, it's like 16 August. So they found like the stem cells have their own memory. Then the Alzheimer. So this is also recent. So they are finding the ESCs where they were differentiated into neuron progenitor cells. So these are again the basically the stem cells. So these were differentiated into neuron progenitor cells in vitro, and these cells were transplanted into rat models. Obviously, everyone knows like we are using mouse for research, right? Because 99.9% of the DNA of the mouse and humans are same. So yeah, then the SC treated rats, they perform, that is the stem cell treated rats, they perform better at cognitive tests. Cognitive means the test where they need to use their brain. So for example, they need to fetch food or something, so they need to use their brain to hunt for food, right? So like that. So then the bone marrow mesenchymal cells were also shown to reduce the AD causing a beta deposits in the brain. So if you know that Alzheimer, it's basically there is this deposits of proteins in the brains, that's why people get Alzheimer. So using this embryonic stem cells, they are now finding a new alternative for Alzheimer disease treatment, which is interesting. Let's move to gene therapy. So it's basically a treatment of disease by transferring of genetic material into the cells. So this is called as gene therapy. Okay. So it's basically a treatment of disease by transferring of genetic material into the cells. Now, it's basically a technique that modifies a person's genes. Okay, now you're going to modify a person's gene. That is, you're going to change the genetic makeup of the person to treat or cure a disease. For example, a gene is not functional, then you will either delete that gene or you will replace that gene. Okay, so the gene therapies can work by several mechanisms. So first is like replacing a disease causing gene with a healthy copy. So you replace, for example, this gene is not good, if it's causing some mutation in the person, then you can delete this gene and you can put a healthy copy of that gene. Or second is you can like directly inactivate, you can shut down, shut off the gene or disease causing gene because it's not functioning properly. And third thing you can do is you can either like introduce a new or modified gene into the body to help treat a disease. Okay. Now, so these are the varieties of the gene therapy products which we use. So first is like we use plasmid DNA. Okay, so we can put the gene we want. For example, there is a person who has whose gene is mutated, and he needs that, he got some disease due to that. So what we do is we can use bacterial plasmid DNA. So these are the plasmids which are present in bacteria. So we can add a healthy copy of that gene into the plasmid, and then we can put the plasmid in the person's like the cells, and once the plasmid get attached, the healthy copy of the gene will get replaced. And then we can use viral vectors. So we can use actual viruses, inside which you can put the DNA, and then this virus can go and infect the human cell, and then it can replace the mutant copy with a healthy one. Then obviously we have the bacterial vectors as well. Then the human editing technology, which is the CRISPR, right? So obviously I'm going to discuss about CRISPR in the further slides. And we have the patient derived cellular gene therapy products. So again, this is different. So this is the CRISPR model in this picture. There are two types of gene therapy, ex vivo and in vivo. So in vivo, we use non-viral delivery system, which means it's in a name, right? We are not going to use virus. We are going to use nanoparticles. So these are the nanoparticles inside which there will be DNA, okay? Or else there are, here you can see like basically inside the, like there is a DNA and there is a myelin sheath. So we can either use nanoparticles, or there are also I guess lipopolysaccharide or something, I'm not sure, but yeah, there are different types of non-viral delivery systems. And obviously second is the viral delivery system, where we use actual viruses, we put DNA inside them, and they'll go and infect the cells of the body, and then they can replace the gene. So this happens in vivo. In vivo is inside the person. Now, what is ex vivo? Ex vivo means in the lab. So it means that what we are going to do is we are going to take the cells from the patient. Okay. Now these cells are going to be plated on a plate in the lab, and in the lab we are going to modify these cells. And now when these cells are healthy, that is the modified copy of the, the healthy copy of the gene is incorporated. We are going to put these cells in back into the patient, and now these cells will divide, and it will basically, the patient can will feel better. So this is what the gene therapy is all about. So CRISPR is also called as the clustered regularly interspaced pattern of prokaryotic organisms such as bacteria or archaea. So how we have immunity, like if we get like I don't know, fever or something, and for example bacteria, for example, like I don't know, it evades us, and then we get cough, cold, something like that. Then the next time the infection wouldn't be that severe. What does it mean? It means like our body now has gotten stronger. For example, let's talk about the COVID thing. So during the COVID, the first wave, like people are not, acclimatized to the COVID virus, right? But after some, like after a few months, there was herd immunity which was formed. So now the people are not getting that much sick, and now it's the COVID is almost like just like another flu. So which means that the body is now like preparing to fight back, right? The body is growing its own immunity. Similarly, bacteria or archaea, okay, which is also the prokaryotic organism, bacteria, and then the archaebacteria. So these bacteria, they also have their own immune system. So these are the what I want to point out is these are the viruses which infect the bacteria. How bacteria infects us? Similarly, there are viruses which affects only the bacteria and not the humans. So these are called as the bacteriophages. Okay. So these viruses are called as bacteriophages. You can see it literally looks like a spider. So it sits on the bacteria, and then it kind of punctures a hole in the bacteria. And what it does, the DNA of the bacteriophage goes inside the bacteria. And once the DNA is inside the bacteria, it starts infecting all the bacteria from the inside. And this is the basically the transmission electron microscope image. You can see, you will literally see the bacteriophage sitting on the bacteria, right? So what happens is, obviously, as I said, like, body is getting immune. So what does the bacteria cell is doing? Once the DNA is getting into inside the bacteria, the bacteria is ready to fight it. So the bacteria has its own what we say, the proteins which can actually, like, chop off the viral DNA which is coming inside. So these proteins are called as the Cas. So you can see here the Cas proteins, it's called as the CRISPR associated proteins. Okay, so these are called as the Cas proteins, which are the endonucleases. So it acts literally like a molecular scissor. It chops off the DNA. And what does it do? It doesn't throw everything away. It chops the viral DNA which is entering, and a certain part of the viral DNA it takes and it attaches to its own genome. Now imagine, like, after every for example, a month, the bacteria is getting evaded by some other virus like that. Now it has, like, chopping off the DNA and inserting a certain part of the viral DNA into the bacterial genome, and now it has formed. So this we call as the clustered regularly interspaced palindromic repeats. So this is basically a memory of the subsequent infection. Now if the virus of the same type attacks again on the bacteria, then the bacteria is ready to fight. Why? Because now it has this copy of the gene of the this virus, which was because it had infected earlier. So this is how it basically, CRISPR works. I'll explain it in further slides as well. So these sequences are derived from the DNA fragments, okay, of the bacteria, and that had previously infected the bacteria. Okay, they are used to detect and destroy DNA from similar bacteriophages during the subsequent infections. Now these are the lytic and lysogenic cycles. So the bacteriophages, which you saw here, they form like two parts. They can either go into the lytic cycle or either lysogenic cycle. What does it mean? Now imagine like a bacteriophage. It comes, it sits on the bacteria. Okay, it bores a hole in it. It injects the DNA material. So here you can see it sits on it, it injects the genetic material. Now it has two things. It will attach to the genome. You can see the red part and the blue part, so it attaches. Now there are two things. It can either start making the proteins of the bacteriophage, because there is transcription going on continuously, right? So if it will also start making the bacteriophages protein, and once it start making this protein, will reassemble and make thousands of bacteriophages, and the cell will like swell up at a certain point that it couldn't, like, it can't hold more, and it will just burst out, leaving all the viruses into the surrounding. Or else what can it do? It can remain in a dormant state. Like, now it has infected this, now it will go, it will divide the plasmid, like for example the genome, it will divide into two, and then the cell will split up. Now one cell will become 2 cell, 4 cell, 8 cell, and the bacteriophage genome will divide simultaneously. And once it thinks, like, okay, now I need to get out, it will go into the lytic cycle, it will start making the proteins, and the bacteriophages burst out. So this is what bacteriophage does to a bacteria. So this is the regularly interspaced RNA. So we have this, as I told, there's literally patches of DNA which are there. So these sequences, they play a key important role in the antiviral or the antiphage defense system of the prokaryotes and provide a form of acquired immunity. So the CRISPR, it's found in approximately 50% of sequenced bacterial genomes and 90% of the sequenced archaea. So you can see, like, so like there's so many sequences of bacteriophages which are inside the bacteria. So the bacteria is always ready to fight back. Then the Cas protein. So these are the proteins. It's called CRISPR associated proteins. So they literally act as a molecular scissor, and they chop off the DNA. Here you can see in the diagram. Literally chop off the DNA, and then this is the viral DNA. The red one is the viral DNA. Here you can understand more. So imagine like a virus is entering in the bacteria. Now the virus is inside a bacteria. Okay. Now what happens is the Cas protein, it recognizes, oh, this is it's a virus, it's a viral DNA. It's not our DNA of the bacteria. It will go and it will chop all the DNA and take only one fragment of it. Now the Cas protein will carry this one fragment. Okay. And it will go and it will paste it in the bacterial genome. Okay, now it has pasted this in the bacterial genome. Now what happens? Now this bacterial genome, it will form a RNA copy. Obviously, there will be transcription, right? They are going to form RNA copies from. Now it will form a RNA copies, and these RNA copies will be bounded to a Cas9 protein, right? And this literally, like, it would act as a scout. It will go from like left, right, like that. It will act as a scout, and as soon as it thinks, okay, same type of virus is attacking me again, it will go and chop it off. Why? Because now it knows, because I already have a copy of it in my genome. So it will go and chop it off. That's how it basically works. So there are some phases. So there is CRISPR, so we call it clustered regularly interspaced short palindromic repeats, and a group of scientists. Okay, so there is a co-founder, Dr. Emmanuel Carpentier, and also Dr. Jennifer Doudna. So they both got Nobel Prize for this CRISPR in, I guess, 2020, if I'm sure, I don't know. Yeah. So in 2020 they got this Nobel Prize, okay, for finding this CRISPR. So what they did was, so using this bacterial system, what they did was they thought like maybe this chopping mechanism can be very specific and it could be designed for humans as well, to to like to, for example, knock out a gene or knock in a gene. So it can be used for humans as well, and they did the research in the bacteria on the CRISPR. Then next thing is Cas9, which we saw, the endonuclease. It's called as the CRISPR associated Cas9 endonuclease or the enzyme, and it acts as a molecular scissor. Then the DNA, we know that, RNA, we know, and then there is the guide RNA. Okay, so this is very important because guide RNA is the one which is going to go and attach to the Cas9. Okay, and then it, as the name suggests, guide RNA. So it's going to guide the Cas9 to come and sit on the DNA and cut it off. So I'll show this. Yeah. So this is the guide RNA. So here you can see it literally represents like a map, right? So it goes like, it basically forms a complex with the Cas9, and then it tells the Cas9 to, okay, now come here and then, like just cut this part off. So the guide RNA is made up of two parts. So it's called as the CRRNA, which is the target specific, okay, and the tracer RNA, okay, TRACR tracer RNA. So the guide RNA is basically two parts of this. Now what happens is, now what did Jennifer Donner did? They modified this part, the CRRNA sequence. They modified this part in the guide RNA, and now what the guide RNA did? Guide RNA started cutting the complementary parts in the DNA. Okay. Now it went. So for example, if the CRRNA is complementary with this sequence, okay, in the DNA, it will go and sit, it will be complementarily bind to it, and then it will chop it off. The Cas9 will chop it off. So there are two parts, the Cas9 and the guide RNA. Guide RNA helps to guide the Cas RNA to cut the DNA. Now how does it cut the DNA? So it is double stranded cut. What does it mean? It will cut both the strands of the DNA, not just one. So it is a double stranded cut. Now if, obviously, there always be something in our cell, right? We cannot let the DNA be damaged, right? So once a DNA is cut, there will always be DNA repair mechanisms in the cell, which will always be ready to like make up everything. Like for example, they've cut something, then there will always be a mechanism, a repair mechanism. So there are two types of mechanism. Okay, one second here, where did we go? I haven't. Okay, anyways, there are two types of mechanism. There is HDR, which is the uh, homology directed repair, and then there is NHEJ, which is non-homologous end joining. So what does it mean? Like now, if it is cutting both the strand, it will randomly just like it's non-homologous. It wouldn't mind. So it just joins it, and the gene will be inactivated. Okay, so there are two mechanisms. So the Cas9 protein forms a complex with the guide RNA, right? So the Cas9 endonuclease, it forms a complex with the guide RNA. The complex attaches to a matching genome. If the guide RNA is complementary to the DNA, okay, then the Cas9 will chop it off, and then this programmed DNA may be inserted by the cut. So if you want, like I don't want the gene to be inactivated, but I want to replace it with a healthy gene. So what will I do? I'll just put a nice programmed healthy copy of the gene in the environment, and once the Cas9 cuts it, then the healthy copy, it will go and sit in the cut region. So now the mutant copy is replaced by a healthy copy, and this is what happens inside a cell. Then let's think about the PAM sequence. So how does the like guide RNA know, like from where to, sorry, the Cas9 knows from where to start cutting? So this is where the PAM sequence comes in. So PAM is basically protospacer adjacent motif. It's called as PAM as short, and it is very small. It's like 2 to 6 bases in length, and that follows the DNA region which is targeted for cleavage by the CRISPR system. For example, if the now see, here is the guide RNA with the Cas9. Now the guide RNA sees, okay, so this is the complementary thing. Okay, so after seeing the complementary, the Cas9 will see, okay, where is the PAM sequence. If the PAM sequence, if the Cas9 sees, okay, this is the PAM sequence, it will cut two, three, like say 3, 4 nucleotides downstream of the like the PAM sequence. So every time, whenever you cut something with CRISPR, you'll always have to see where is the PAM sequence. If the PAM sequence come in between your gene of interest, then the gene of interest might get cut. Okay, so when you're always designing the guide RNA for your experiments or something, you always need to make sure like where is the PAM sequence. And here is just like a examples. So for example, in streptococcus pyogenes, you can see that there is NGG PAM sequence, right? So it's like 3 to 4 nucleotides long. N is basically any nucleotide. ATCG, you can place any nucleotide in it. Then what are the applications? So you can either disrupt a gene using CRISPR, you can either delete the gene using CRISPR, or you can correct or insert a new gene using CRISPR. So you can see like how powerful this gene editing technology is. You can use it in any system. You can use it in fungus, you can use it in bacteria, in humans, anything. Then what are the applications? To knock in, knock out, to create point mutations. If you want to activate a gene, if you want to repress a gene, now, for example, if you want to image something, you don't know if that gene is present, so you can image something, you can use CRISPR. Okay, so you can basically use guide RNA and Cas9, and then you can place a fluorescent molecule like GFP or something on the guide RNA, and once it go and attaches to the DNA, it will just it fluorescent, so it fluoresces and you will know, okay, like that gene is there. So you can do the imaging also. You know that the genes are present, so you can do imaging also using the CRISPR fish is basically the experiment basically called that. Then you can develop a customized gene therapy for patients. So you can select, it's amazing. There is a recent research in CRISPR. So there is stem cell based functional genomics for the large scale analysis of genetic variants. Um, what I so one of the yeah, so this paper is amazing. So here what they did was they kind of genetically modified the mosquitoes. So they somehow, like using CRISPR, they deleted the gene which basically was complementary to the malarial parasite, and they deleted this gene, and now the mosquito cannot harbor the malaria virus. So once a mosquito even goes and bites a human, it will not transfer the malaria because it doesn't have it. So it's amazing, like they use CRISPR to design these mosquitoes, so like people will not get malaria like by biting this. Then second, you have this peptide mediated CRISPR engineering of cells. Then so they have, it's basically the hardwired independent peptide mediated delivery method for the CRISPR mediated engineering of T cells. So that's also amazing. Yeah, I mean, you can just go through these links I've attached here, so you can just go through these papers, and it's nice. So using the CRISPR in lab, so imagine like you have a plate full of stem cells and everything. Then finally, so we have the PAM set. As I told, the Cas9 is going to break two strands, so it's going to be like it's going to have two repair mechanisms in the cell. There is NHEJ, non-homologous end joining, and there is HDR, homology directed repair. So you can see it's just just joined, it's getting joined while creating some mutation in between, so the gene is not functional anymore. And second is HDR, so where you can literally insert another gene, homology directed repair. So yeah, and then what you're going to do, where you get a plate, you put all the plasmids. So there will be one plasmid which will have, which is a basic donor plasmid, which will have ampicillin resistant gene. Okay, and there will be second plasmid which will have the Cas9 gene, and whatever you want to insert that gene. And third plasmid is a GFP plasmid, so you will know like which cells have taken up the CRISPR Cas or not. Then you put everything in one mixture, in one tube, and then you use electroporation. Now, like you can use the calcium thing also. Like for example, you can use calcium ions to make holes in it and then put the plasmid inside, or you can use electroporation. It depends upon like what you have in the lab. Then you can use electroporation. So this electroporation will bore holes on the cell wall, and all of these plasmid now can go into your into the cells basically. And then you can visualize this using the microscope. Now this you can see, okay, because you have added the GFP also, right? So you can see, okay, now the which cells have taken the plasmid. Now positive also. What does it mean? Now there can be cells which haven't at all taken up CRISPR-Cas plasmid, but they have only taken up GFP. So it's the cell, even these cells are useless, right? We want cells to take up the Cas9 plasmid, because if the Cas9 plasmid is not taken up by the cell, then the Cas9, I mean the CRISPR-Cas protein complex wouldn't be formed. And if the complex is not going to be formed, then there will be no chopping of the DNA inside the cell. So we don't want this false positive. So what we do FACS sorting. So here we sort all the good cells, and basically we throw out all the bad cells, and then we get a nice colonies which are all of like full positive, proper positive cells. Then we, like for example, if this is one colony, this is one colony, we plate one colony in one cell plate basically. So we put one colony, like individual colonies, and as the colonies grow more, we basically split them up. We take more plates, and then we can use PCR to check if there is a knockout or not, or if there is knock-in or not, like that. And then we can if we think, okay, no, there is a good band, and it means like maybe the gene is knocked out and you still need to be sure, then you can send the sample for sequencing, and this can be done by Sanger sequencing or something like that. And then you can see, okay, now so basically these are the Sanger sequencing. You can see every peak, it is a different color. So green represent A, or this orange thing represents T, black is G. So this peaks, so if the peaks are like more, if the peaks are nice, it means like the results are more reliable. But if there are like overlapping peaks, you can see here, it's like very overlapping, and like it's not good peaks, then it means like the results are not reliable. And yeah, and then you see like what the how is the gene knocked out or not. So this is the last part. This is just like 3-4 slides more. So this is basically, I worked on this project during my lab rotation here at LMU Munich. So this is like a part of my master's program. So I have a 2 years of master's program, and we have compulsory lab rotation. So during one lab rotation, I worked on this embryonic stem cells, and I used CRISPR-Cas for this. So what my work was is I worked on this transposable element. So if you have heard about jumping genes, which literally means like there are some like genes in the, I mean the parts of the DNA which randomly breaks off and attaches to like another part in the genome. So and even humans have that. They are called as the ALU sequences, ALU. So these transposable elements, they can be harmful, right? They can cause insertional mutagenesis, they can cause like transcriptional perturbations, genome instability. So to avoid this, these genes need to be silenced. Like they need to be silenced. They should be switched off, or else there will be a lot of mutations in the DNA. So this is one of the enzyme called as the SETDB1. So it is called as the set domain bifurcated histone lysine methyltransferase. It's a long name. So what does it do? Simply is its work is to like take a methyl group and attach it to the DNA. So once a methyl group is attached, once a methylation is done, the gene is switched off. Okay. So this is what it does. So my work was to study the interaction of these proteins with this SETDB1. Okay. Now to study the interaction of this protein, I need to first create a knockout cell line, right? So for this, one second, I just, okay. So for this, what I did was first I took this cell plate. Okay, so I took this cell plate. I treated it with two plasmids. So first was the plasmid which had eGFP, is basically a modified form of GFP, and puromycin. And there was another plasmid which had proper sgRNA plus Cas9. Because what does this do? This is basically going to form the protein. CRISPR-Cas9 protein, proper protein, which are going to go and chop it off. So this is very important. We need to make sure that this plasmid is inside the cell. So I made different controls. I saw in, obviously I use the microscope, I visualize the cells, everything, and then I know, okay, like, like the cells are good, and like they are showing like a proper, like, proper signal. Then what I did was I took the colonies and I picked the colonies and put it in as I told you, right? So one colony in one like cell plate. So like one well. So like one colony, one well, like that. So after this, the cells will grow. There will be more colonies. So after a certain number of colonies or the cells are reached, we go to DNA extraction. So we extract the DNA, then we use the PCR, we amplify the gene which we want. Okay. After amplifying, we purify the PCR product and we check it using the gel electrophoresis. Now, if we think, okay, the gel is fantastic, you got the bands that you want, you can send the colonies for Sanger sequencing. On simultaneously, you can also check the DNA concentration using nanodrop. Now if you for like for example, during my project, what happened was during Sanger sequencing, I was like, I mean, there is a knockout, but it is not a complete knockout. I think like two colonies somehow mixed up. So what I did was I used blue-white screening. So blue-white screening is a bacterial technique where we put single strand of our gene into the plasmid of the bacteria, and then we plate it. So the white colonies are the ones that we want, and blue colonies are the ones which doesn't have the plasmid. So I took these white colonies and again sent them for sequencing. And once I got a proper knockout, I did the protein validation studies. And yeah, so this is like a last part. So this is how we basically maintain the cells in the lab. You take the plate, you basically like take out all the media, the red color media it has. You take everything out. You put PBS. PBS is like a washing thing. So you wash the cells. Okay, you put 3-4 ml of PBS. Then you mix it up. You remove the PBS. Then you put 1 ml of trypsin. What is trypsin? Trypsin is a protease. So as I told you, the cells have a tendency to attach, right? So trypsin will detach the cells and the cells will start to float um, in the PBS. So after putting the trypsin, the cells will, if they have like clumped, they will separate. So you will have like a single segregated cells. And you can take these cells, you put it in the falcon tube, centrifuge it, and all the cells will be at the bottom. Okay. And then you put like 50 microliter of the media in another small tube, and then you count it. And like this is how you basically maintain and count the cells. And then you can, once you have counted the cells, you take 1 million cells and plate it in another plate. So this is how you basically passage the cells. Passage in the sense you divide the cell plate. And yeah, so this is how it is. And yeah, thank you so much. I've spoken a lot, and somehow I'm on time. Okay.
Thank you, Ms. Mrunal, for giving your valuable time and speaking in today's webinar. I, on
Yeah.
behalf of the whole cyber team, really appreciate and thank you for today's session. And today's webinar was really encouraging and motivating for all of us, and we would like to attend more of such webinars by you in the future. Thank you.
Thank you.